Hello, everyone. Today I have the pleasure to hear an opinion on FIDA from Prof.Stefan Rüdiger from Utrecht University, who will tell us his opinion on FIDA in usage for protein chemistry of disease.
Thank you, Maya. My laboratory is actually working on protein folding and aggregation and the protein quality control system controlling protein aggregates. And that is actually the application we use FIDA for. And I would like to briefly illustrate that in a short presentation. What we do find, for example, the disease of Alzheimer. We have protein aggregation outside the cell, where there are beta 42 aggregates, and we have protein aggregation inside the cell, where the protein tau aggregates. Now, our lab would like to study this aggregation process, and that means we need to study and understand how actually fibrils grow in size and also shrink in size, and that's depending, for example, on the action of molecular chaperones. And so for us, actually, the question was, how can we actually quantify that process, which means determine the size of the protein fibrils. Now, when we look at the process of aggregation, then we do have monomers that slowly grow into oligomers, and those slowly grow into fibrils. And basically, when thinking about how can we develop a strategy to remove such fibrils, I mean, it would be an option to think about having a compound that would target specifically oligomers or fibrils, but actually not the monomers, so fibril specific, right? So that would be the overall goal. So for that, particular would be very interesting to focus on the earliest fibrils. Now, but then we need to be to be able to do that, we need to be able to measure that. And so one prerequisite to measure that is that we need to develop a compound that specifically recognizes oligomers or fibrils. And that actually we succeeded. We developed a peptide, which we call fibril paint, that, as you can see here, specifically binds to fibrils, but not to the monomers. And this is a peptide enriched in aromatic residues, tryptophan, histidine, or actually arginine, and these are all residues that can do pi stacking interactions. So, good. But now we have a peptide that actually binds to fibrils. But how actually can we now measure the binding to fibrils? And that was a tremendous challenge for us to get this done. And the solution for us was actually that we now actually employed FIDA. And what actually FIDA does and allows us to do is that you send your sample through a capillary, and you get a diffusion profile. And that diffusion profile depends on the size of the sample. So a small peptide would have a very sharp profile, while a larger sample would have a much broader profile. So a fluorescently labeled peptide, such as this fibril paint compound, would be small. If the fibril paint peptide binds to the larger fibril, you get a broader profile. And what the FIDA does, it measures basically the diffusion from out the parabole actually towards the sides of the capillary a sample is shot through. And based on that, you can calculate the diffusion compound, the hydrodynamic radius based on the diffusion. And that correlates with the apparent size of the of any particle you look at. So when you have a peptide alone, you get a sharp peak. When the peptide binds to the fibril, as is visualized here, you get a broader peak. Now, good. And with that, we could now look at protein fibrils. For example, fibrils of the tau protein that aggregates in Alzheimer, right? So you see here on the left, the structure of tau in Alzheimer. And then you see here on the right, the hydrodynamic radius actually for fibril paint alone, for the monomer, and for the fibril. And what you can see here is that the hydrodynamic radius actually of the fibril is around 40 nanometer. The peptide alone is very small, and there's actually no interaction with the monomer at all. So, and that gives a pretty clear readout for the size of the tau fibril. Now, tau fibrils, you get in Alzheimer, but you think, okay, in other disease, you have other fibrils that aggregate, right? So, and what about those? So we also tested that on the fibrils of the beta protein that also aggregates in Alzheimer, of alpha-synuclein that aggregates in Parkinson, and also of the Huntington protein, for which we now don't have a cryo-EM image. Yeah. And actually checked, okay, can we also measure those in the combination with fibril paint and FIDA? And the results of this is that you see here, actually for the 4 different proteins, the size of the aggregates, and you can see they're typically around 50 nanometer in hydrodynamic radius. The Huntington is longer in our hands. It aggregates extremely fast. So that allows us actually to get a quantitative impression on the fibrils. Now, these are actually artificial fibrils. There are other methods, like we now use this fibril paint peptide. So there could be other methods. You can directly, for example, label fibrils, build up fibrils out of labeled compounds. But actually, one goal we have is to measure the size of fibrils in patients. And these fibrils are not and never fluorescently labeled. So we need to employ something that allows us to label these fibrils. And so we also employed fibril paint to measure the size of patient-derived tau fibrils from Alzheimer's disease, from Pick's disease, and from corticobasal degeneration. And what you can see is that these are relatively large fibril structures, while the artificial fibrils down here are relatively small fibrils. So now, when testing actually the FIDA on these various fibrils, you can see that we get from all also these patient-derived samples, we get actually an accurate hydrodynamic radius. Now, question is, this hydrodynamic radius is when we want to think, good, it's a very useful parameter, but what does it actually mean, right? So what we want to have actually the length of the fibril. So what we want to have is a ruler for our protein fibrils. Yeah, so you see here on the image, you see a negative stain EM picture of the PRT helical filament fibril, patient-derived fibril of Alzheimer. So good. So what we did is that we actually predicted the hydrodynamic radius of these PRT helical filament structure with for increasing lengths of fibrils. So you see on the right, you see the fibril structure, right, of the Alzheimer PRT helical filament structure as it was solved by cryo-EM by Shorshera's labs. And if you know the structure of a particle, there's a FIDA software module that allows you to predict the hydrodynamic radius of a structure of a certain size. So, and with that, we could actually now predict what would be the hydrodynamic radius for increasing fibril length. So, and we know from these tau fibrils that the distance between two layers is 4.7 angstrom. And then you, for example, know 212 layers would be 100 nanometer. So, and if we, such as the data I've shown before, for the tau fibrils, we have an hydrodynamic radius of 40 nanometers, we can calculate that this corresponds to 900 layers, or actually 425 nanometer. So, and this is actually quite useful information because this gives you it's an ensemble value. It gives you an ensemble value of your sample. And now we correlated that with electron micrographs we had from the same fibril. So actually, Françoise Decker in lab, she actually counted the length of the paired helical filaments in our TEM pictures. Yeah, and you can actually recognize in the paired helical filament structures, you see the repetitive twist of the fibril, and that actually allows you pretty much to identify fibrils that lay nicely on the grid, and you can estimate the length of those. And and Françoise actually counted 48 PHF structures, and she found that they have in the sample an average length of 430 nanometers. So our FIDA value had been 425, which is an amazingly good correlation of both techniques. So, which means if I summarize that in the nutshell, and that's actually what we use actually the fibrils for, is that we actually now have actually a molecular ruler for protein fibrils, and use a combination of actually the fibril paint peptides, so you see on the left, and the application of these fibril paint peptides with fibrils in the FIDA. And yeah, this is what I have to share. And I, of course, don't do that alone. I'm supported by a fantastic group of people, which I also would like to thank. And if you would have specific information on the FIDA, these are also people who can give you advice.
Wonderful. Thank you so much. That was a captivating presentation and a very nice picture. Thank you so much. I have two questions to you. So first one is, what would you miss the most if FIDA was to just magically disappear from your lab?
Well, we have the door of the room where the FIDA is locked. I mean, I should say it is just to make sure it doesn't happen. Although I should say it's widely available. We have it now. It's in a protein research center, which is used by all the life science group in Utrecht. But FIDA is actually the only technique right now that allows us to measure the size of protein fibrils, yeah, and also the monitoring of the length and increase or shrinkage of fibrils. So, for example, by the action of molecular chaperones, by sonication, or whatever. And because we can so specifically label the fibrils, it also works in dirty solutions. Yeah, we can do that in blood plasma or in lysates. It's super specific. And you can distinguish with that, we can distinguish the fibrils from any other type of aggregates. Yeah. And with this means that the entire monitoring of protein fibrils, we couldn't use that. We couldn't simply do that without FIDA.
Mm-hmm. Well, then it's good you keep the door closed. But if anybody, like, it's good to spread the knowledge that it is open also for use for other departments. That's amazing. And my other question is about the future. So what plans do you have for applying FIDA in the future research, in research that is to be published yet?
Yeah, so we actually now would like to see that can we actually target protein fibrils for degradation, and for that we use now also fluorescently labeled ubiquitin. And what we can see now also in the feeders, we can follow the entire ubiquitin process. So traveling of the fluorescent labeled ubiquitin from the E1 ligase to E2 to E3 and onto the fibril, and you can monitor that because for every ubiquitin ligase, you can predict the apparent dynamic radius. And then you can see that the ubiquitin, actually moiety, is based on the size of the particle, really goes from E1 to E2 to E3 and ends up on the fibril. And that is what we are now exploiting to see, can we also study with that and the ubiquitination and potentially degradation of protein fibrils.
Wonderful. That sounds like a very exciting plan for the future. And thank you so much for your opinions. And if anybody has any questions, you're always welcome to contact us. And yeah, I wish you a great day.
Yes, thank you so much, Maya.